Thank you, and welcome to the second session of the New Medical Frontier. It's nice to have you here, and we hope we have a very stimulating morning for you, both in the lecture and in the Q&A that follows. I'm John Ferries, co-chair with Pete Bleiler of the series, and we're very grateful to all of you who are on the committee that have helped us put this together. A key thing I want to announce, or at least make you aware of it if you don't already, these note cards you received, you fill them out with any questions that you have during the presentation, and then give them to one of us at the front during the break, because it will be sorted out by our moderator who will be conducting the Q&A, and those will become the basis for the questions that she asks. Before going any further, though, I want to give some credit to the people who have been our underwriters for this entire series of the New Medical Frontier. Caldwell Law, founded in 1994. Caldwell Law assists clients in estate planning and settlement, elderly law and advocacy, long-term care planning, fiduciary services, and trust administration. Our friend nearby, Dartmouth-Hitchcock. It is a nonprofit academic health system that provides access to more than 1,000 primary care doctors and specialists in almost every area of medicine, delivering care in Lebanon, New Hampshire at its flagship, Dartmouth-Hitchcock Medical Center, and at 24 ambulatory clinics and 4 affiliate hospitals in the region. The Jack and Dorothy Byrne Foundation, providing financial support to a wide range of nonprofit organizations focusing on academics, students, adolescents, children, and youth, and economically disadvantaged people. To Kendall at Hanover. Located in Hanover, Kendall offers a vibrant life plan community for independent living and person-centered continuing care for assisted living and nursing care. And Wells Fargo Investment Advisors. Wells Fargo Advisors and online tools are supported by some of the nation's top investment research analysts and strategists with a history of serving communities across the nation. And those are the underwriters for the series. Today, we also have a sponsor for today's session, and that is Coldwell Banker Redpath and Company, covering both New Hampshire and Vermont. Coldwell Banker Redpath has more than 40 years of experience as a real estate broker, assisting clients purchase, rent, or sell their home, and operating with integrity and exceptional service. I'd like to hopefully have us give now a round of applause to these wonderful underwriters and sponsors. Thank you. It is now my privilege to introduce the moderator who will introduce the speaker. In each session, we have a different person as moderator. Their role is to introduce the speaker and then to conduct the Q&A. This year, the person who's going to introduce Dr. Eric Green is Sylvia Paxton. And for those of you with Osher, you know that Sylvia is the current president of Osher at Dartmouth. It is with great warmth that I bring forth Sylvia Paxton.
Thank you so much and welcome. And before I introduce Dr. Green, I just want to take a moment to remind all of us that events like this don't happen in a vacuum. John Ferries and Pete Bleiler have a wonderful committee who have been listed in the program. But if you look around, you see people at the door who are collecting tickets, people pick up cookies, people pass out cookies. We have our Osher staff, and we have the wonderful staff here at the Hopkins Center, and I would just like to say thank you to all of you. So when I was given the privilege of introducing Dr. Green, and I went online to learn a little bit about him, I learned that he had earned an MD and a PhD in 1987. And having 2 kids who've earned PhDs and knowing the time it took for them to do that, I asked him, how could you possibly do that? And he told me that now there's a real effort to make sure that researchers have clinical experience as well. And there are programs with medical schools that combine the two. And I have threatened, but I'm not going to do it, to call you Dr. Green. Eric grew up professionally, with a family, by the way, that are scientists, in the genome era and has been on the cutting edge of genomics research for almost 30 years. His appointment, he has been appointed as the director of the National Human Genome Research Institute in 2009, part of our National Institute of Health. This Genome Research Institute is the largest organization in the world dedicated to genomic research. He has said, quote, "My job going forward is to push the application of genomes into all areas of biomedical research beyond the National Genome Research Institute's boundaries and find effective ways to collaborate with a wide range of researchers to translate genomic discoveries into medical advances." I took a wonderful course which Osher offered called Designer Genes last fall. Ian Sim is here, and I want to just remind you, if you're fascinated by this, it's being offered again in the fall catalog. One of the members of that class said, Benissa Saint-Domingue said, "I think this is the most exciting time in the history of man to live because of all these incredible discoveries and the possibilities and the hope of genomics." So I introduce you to Eric Green, who's going to inform us all about them. Thank you.
Well, thank you, Sylvia, for that very kind introduction. Good morning, everybody. It's a pleasure to be here in this beautiful part of the country, and I genuinely look forward to spending the morning with you, both giving you an overview now of this exciting area, but also to hear what you are interested in talking about during the question and answer session, and I think it's going to be fun to have that interaction. Recognizing that there's a diversity among you in terms of your knowledge and background familiarity with DNA, with genetics, with genomics, I recognize it was probably important to start with some basics before I build on the story that I want to share with you for the next 50 or 55 minutes or so. And let's just start with some very simple concepts so you can fully appreciate the story I'm going to unfold for you this morning. The human body consists of something on the order of 10 trillion cells or so. But interestingly, every one of those cells operates off the identical blueprint. Whether it's a brain cell or a muscle cell or a liver cell, it's still operating off of the same fundamental blueprint information for creating that cell, for operating that cell. Now, we've learned a lot in the last 100 years or so about what it is about the human blueprint. It sits in the middle of the cell in something called the nucleus, but more importantly, it's housed in structures called chromosomes, sort of the suitcases that carry this blueprint information from cell to cell, from generation to generation. But the real revelation about our blueprint, which came in the 1950s, was a recognition that it was DNA. It was this molecule called DNA, complicated name, easier to abbreviate as DNA, that really is the blueprint information for all human cells, in fact, for all living creatures. Well, the 1960s and the 1970s, and then a little bit into the 1980s, brought remarkable advances scientifically in being able to study this blueprint information, this DNA. We learned how to manipulate DNA, how to isolate it, how to clone it, how to propagate it in the laboratory, and eventually how to read it. And it turns out that DNA is incredibly complicated, except by composition, it's incredibly simple. It really only consists of 4 different chemicals, and the easier way to think about the chemicals is just by their first letter, G, A, T, and C. And it's the order of the chemicals across these stretches of millions and millions and millions of letters that encode all the information necessary for life. And as we learned how to read the DNA and be able to determine the order of the G's, A's, T's, and C's within DNA, we begin to recognize that, wow, we may be able to go through and read out and understand our fundamental blueprint. And part of the reason we got quite motivated about this in the 1980s and the 1990s in particular was that it was a finite problem. The human blueprint consists of 3 billion of these bases, or letters. 3 billion G's, A's, T's, and C's in the precise order is all the information necessary for creating and operating a human body. Well, this led to the notion of, could we comprehensively study all of the DNA of humans and other organisms? And all the DNA of an organism is called a genome. And this gave birth to a new field of science in the late 1980s called the field of genomics, the comprehensive study of the DNA of an organism. And in fact, 1987 was the first time the word genomics was even used in the scientific press. In fact, it was in the lead editorial of a brand new journal called Genomics, where the discipline was first named and put into the scientific literature. And they talked about how mapping and sequencing DNA and genomes would have this term called genomics. So keep in mind, the field of genomics has really only existed since 1987, much, much, much younger than other fields of science you may be familiar with, whether it's biochemistry or chemistry or physics or cell biology, all much, much older fields than genomics. By the way, as you heard in the introduction, I graduated as an MD PhD student. The year of my graduation was 1987, which means never once during medical school or graduate school did I hear the word genomics because the word hadn't been invented, which is actually interesting since I'm now responsible for leading the US's institute dealing with genomics. But like all of you, it teaches a very important lesson. We are lifelong learners, and the successful people in this world are ones who embrace the idea of learning for their entire life. And believe me, I've been learning genomics ever since I graduated medical school and graduate school, because it didn't exist as a word. Now, the reason why the late 1980s, why the field was created was because an idea had bubbled up in the scientific community that maybe it was time to read out the complete sequence of the human genome, to completely characterize the human genomic blueprint. And this galvanized around the idea of a project which was launched in 1990 called the Human Genome Project. This was an audacious and unprecedented project that was highly successful and was completed in 13 years, but it was very different than typical biomedical research. It was big science. It involved thousands of researchers, multiple countries all working together in a very coordinated way. It was not the typical way biomedical research had been done up until that point, but it was the right thing to do at the right time, starting in 1990. And in fact, last year we celebrated the fact that it had been 25 years since we had started the Human Genome Project, which was really quite striking for me to think back that it had been a quarter century since the beginning of the Genome Project. I had the remarkable opportunity of getting involved in the Genome Project on day one, October 1, 1990, because I was then training as a pathology resident and was doing postdoctoral research, and just was fortunate enough to be there on the front line and participated from the beginning to the end. To commemorate the 25th anniversary of launching the Genome Project last fall, I had the opportunity to co-author a perspective piece that was published in the scientific journal Nature by the two other people that have led the institute that I now lead. That's Jim Watson, the famous Jim Watson, who discovered the double helical structure of DNA along with Francis Crick, was our first director, and then Francis Collins was the next director. Francis is now the director of the NIH, being appointed by President Obama, and then myself. And we wrote this perspective piece because we really wanted to describe the fact that a quarter century later, since the launch of the Genome Project, genomics has delivered far more than just the sequence of the human genome. It actually changed a lot of the ways that we do big science, big biology. And there's a lot of legacy issues, and some of which I'll weave into my talk, that I think really are a signature of what our contribution has been as a field, even beyond the DNA. It's the style with which we do biomedical research now, and it's proving to be very successful and very important for what needs to be done. So it's been a quarter century of this new field of genomics. I'd been there from the very beginning. I thought it'd be interesting to share with you what I think are the major highlights of genomics over over its first 25 years. The first one I already mentioned, and that was we determined the sequence of the human genome for the very first time by the Human Genome Project. We laid out the 3 billion G's, A's, T's, and C's that represent essentially the code for human life. And that was remarkably successful. But a lot, that ended in April of 2003. And now 13 plus years later, a lot more highlights have been added to this slide. So let me tell you about those and put a little bit of background behind each of them so you can appreciate how much has happened in the 13 years since we've had in hand for the first time the sequence of the human genome. Because it's been remarkable what's happened to genomics, and you heard about some of the things we think about as we try to get genomics being used, not just at our institute and not just here in the US, but actually worldwide in many other applications. And in fact, you may have heard about how genomics and DNA analysis is finding its way into all of these areas here, and they're all exciting and they're all very important. In fact, every one of these areas could represent a talk and a discussion in and of themselves. I'm going to focus my discussion and my highlight reel on applications of genomics to health and disease and medicine, very relevant to this series. And the reason, of course, I'm going to keep it focused on that is where I work. I work at the US National Institutes of Health, where the main focus is turning discovery into health. And I lead one of the 27 institutes and centers that are there, called the National Human Genome Research Institute, which by way of history you should recognize was created by the US Congress to lead the US's effort in the Human Genome Project. But of course, with the Genome Project ending 13 years ago, as you might imagine, we wanted to expand our mission to beyond the Genome Project and now have a much broader-based mission to advance human health through genomics research. And what has happened in the last 13 years, and increasingly with the great attention over the last 5 and 6 years, is to point our ultimate goal of what our institute is trying to do is to enable a new discipline of medicine, if you will, which I will term genomic medicine. And by genomic medicine, I mean using information about an individual patient's genome, their own unique blueprint, to tailor their medical care. Not to treat patients generically, because too often that's what we do, but to recognize each of us has a slightly different blueprint. And if we can enable the use of that blueprint and the differences that each of us have in our DNA, in our blueprint, we could probably be much better at the practice of medicine. And this idea has also been called individualized medicine or personalized medicine. And you'll hear later in the talk, I'll talk about precision medicine. I tend to focus on genomic medicine to exclusively focus on genomic information as opposed to other individualized information that each of us are associated with as well. But how do you do that? I mean, how do you create an ability for a scientific and clinical community to come together and basically start with what the Human Genome Project delivered 13 years ago, an actual blueprint laid out on how humans operate, but then somehow change the practice of medicine, which of course is very complicated. Well, this doesn't happen overnight. It doesn't happen with one step or one project or one effort or one scientist. It is a very long-term project. It involves many steps. We don't know what all of them are. We know what some of them are. And then you have to recognize that it's not going to be a single scientist or a single group that's going to do this. In fact, it's not even going to just be one discipline. It's going to require a lot of creative energies coming together, doing research, making progress, thinking about how to apply it clinically. And of course, it's not a sprint. It is a marathon. And I think that analogy is a very good one to keep in mind. It's thousands of people running shoulder to shoulder from multiple countries, multiple disciplines, trying to figure this out as we go through this journey of a path from the Human Genome Project to realizing genomic medicine. Now, how do you, as somebody responsible for leading a part of the US government that funds this work, how do you sort of organize your thinking? How do you get your community appropriately set and funded for pursuing this long journey towards genomic medicine? And one of the things that we do as a community is to strategize, think about what needs to be done next and what needs to be done to sort of enable the subsequent steps. This worked for us extremely well during the Human Genome Project. And so we used our own playbook to say, well, every time we have an opportunity to think about the future, let's bring the community together, let's strategize, and then let's write this up so that everybody sort of has in front of them a strategic view of what needs to be done. So literally, the day the Human Genome Project ended, in April 2003, our institute concluded a strategic planning process that led to a new strategic vision of what needed to be done starting the day after the Genome Project ended. I was a co-author of this. It was led by Francis Collins. And one of the other co-authors is actually Alan Guttmacher, who's sitting here in the front row, and I'm going to probably bring him into the discussion during the question and answer session. Alan, at that time, was the deputy director of the institute. I had a different position. I was leading our on-campus research program in genomics. And then Alan actually subsequently left our institute to become the director of the Child Health Institute at NIH, where once again we were close colleagues and good friends, both being directors of an institute. Alan was actually trained as a medical geneticist many years, practiced medical genetics in Vermont, and now is a retiree close by to here, which is why he's joined us here today. And then, of course, that by 2010, 2009, we recognized a lot had happened in genomics. I'm going to tell you about some of it. It was time to update that strategic vision. And so once again, we got the community together. Once again, we strategized and published a strategic view of what needed to happen. For the first time, though, we directly embraced this idea of clinical applications of genomics, changing the practice of medicine, enabling genomic medicine, even in the title of this strategic plan. And we also laid out in 2011 a progression, if you will, that you're going to sort of see me play out in the highlight reels, going from doing research to give you very basic understanding about how genomes are put together, to actually how they work, to how genomes are relevant to health and disease, to how you could use genomic information to change how you practice medicine, and hopefully eventually change the effectiveness of healthcare. And these sort of progressive domains become very important organizing framework elements by which we put research projects on to try to see this progression go from the Human Genome Project to enabling genomic medicine. So with these strategic plans as our guiding posts, if you will, for the last 13 years, what else has happened? What are some of the other big highlights of the last quarter century? Well, I would say by far and away the next highlight that deserves significant accolades is the fact that we have significantly reduce the costs associated with reading out the sequence of a human genome. In fact, remarkably, we have reduced the cost of sequencing a human genome by nearly 1 millionfold. Now, let me unpack that a bit more because it didn't happen by accident. And in fact, when Alan and I and Francis Collins and the other co-author Mark Geyer were crafting this strategic plan in 2003, we wrote many things in that, and this was going to be published in the scientific literature with our names on it, and we recognized we were going to have to be audacious and bold, but we probably didn't want to go over the top. But nonetheless, we wrote that there really were needed at that time, 13 years ago, technological leaps that seemed so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. Pretty audacious language, right? And then we gave an example. We really went out on a ledge here, because we gave an example that one of the technological leaps would be the ability to sequence DNA at costs that were lower by 4 to 5 orders of magnitude than the current cost, allowing the human genome to be sequenced for $1,000 or less. Now, why was that so expensive? By the way, we picked $1,000 because we thought it seemed like a bargain. It was about the cost of a CAT scan or a cost of any diagnostic test. Wouldn't that be awesome if we could get complete information about a patient's genome for $1,000? But it was a little audacious because we had literally the day before just completed sequencing the first human genome ever, and it cost about $1 billion, okay? And somehow we were claiming that our scientific community was going to come together and somehow come up with ways to knock 6 zeros off of that figure and deliver the $1,000 genome. And it actually worked. Our institute developed a major granting program and funded scientists around the world, actually, to come up with crazy new ways of sequencing DNA. Some of them didn't work, but some of them did. Meanwhile, the private sector got very interested in this, and major venture capital money came in, and companies were formed. And remarkably, over the last decade, it became a reality. And the idea of a $1,000 genome actually is not quite there, but incredibly close. Shown in green on logarithmic scale, and the graph on the left depicts the cost of sequencing and how it has plummeted. Just gone down remarkably fast and furious ever since some of these new technologies came on the scene. And this has been probably one of the most impressive technological triumphs, at least in the history of the National Institutes of Health, where we had no knowledge that we'd be able to knock 6 zeros off of that figure. We just thought maybe we could, and it's actually been accomplished, and we are incredibly close to having a $1,000 genome. It's also the speed with which we could do it. So I told you that that first human genome that was produced by the Genome Project cost about $1 billion, but it took 6 to 8 years of effort and thousands of researchers involved worldwide having it all come together. But today where we can generate a sequence of a human genome, maybe not quite 1,000, we'll probably get to 1,000 by the end of the year, but somewhere $1,000 to $2,000, a reasonable cost of a test for a clinical test. And we could do it in about a day or 2 or 3, and probably within the next year, it'll probably get down to a day. This has been game-changing in almost all aspects of genomics. Be able to read out genomes of humans that inexpensively has set up all sorts of studies. This is not what I worry about anymore. This has been remarkable. And part of the reason why I'm not worried about it is even the technologies that are sort of shown here in picture form that are being used today we won't even be using them 2 or 3 years from now. There's new technologies coming on the scene that are going to even be better and cheaper than the ones we have. I thought I would share with you, just as an example, this is a new device that is just finding its way into laboratories around the world, actually, made by a particular company. And it uses the latest and the greatest. And it's basically a laboratory on a chip. Here's the laboratory. It's about this big. And literally all you do is put a little bit of blood in a little hole here. And you take this and you plug this into the USB port of your laptop computer. And this device will sequence DNA. Now, it doesn't quite do a human genome in a day, but we're getting there. And it might be that this device or a device like it might be capable of sequencing a human genome in a day for just a few $100s. By the way, this exact device was used in the field during the Ebola outbreak in Africa on laptop computers, not in laboratories, but in tents, where they collected specimens, were able to follow the Ebola outbreak by sequencing the viral genome, which is very, very, very small. It works great for microbes. It's getting better and better, and this might be the future of genome sequencing, not laboratories, but little devices. By the way, what's also way cool about a device like this is that it works equally well in a PC or a Macintosh laptop. So, you can't say that about a lot of other USB devices. So, this is here and now. We can sequence prodigious amounts of DNA, which has set up a circumstance that allowed us to have the third highlight appear in the last quarter century. And that's the fact that we've now not sequenced one human genome, we've now sequenced tens of thousands, and I predict within a year I will change the slide to hundreds of thousands of human genomes have been sequenced. Now, why did we do that? Why is it important to sequence lots of people's genomes? We're not interested in the one genome that we sequence. That just gives us a snapshot of a reference of a human genome. We're interested in how all of us differ, because we are interested in how we vary. This is what our genomes look like, but about 1 out of 1,000 positions in any one of our genomes is different compared to the person sitting next to you or another person you know. We're about 99.9% identical, but that means 1 out of 1,000 letters are different. And we can catalog those differences, but it actually turns out, really importantly, that most of the variants that we have, spelling differences, are actually quite common. In fact, most of the variants you have, somebody else in this auditorium probably has. And so, in fact, that means that if we would sequence enough people, we could catalog the more common variants that exist in the human population. And we want to know that, because most of these variants, and most of your variants, and you have millions of them, don't mean anything. They have no consequence for your health or your well-being. But a subset of them are important. Some of them might not be particularly good variants. They might confer a risk for getting a disease. And others might be good variants. They may protect you from a disease or have some other positive attribute associated with it. But since most of variants are common, wouldn't it be awesome if we could have catalogs of all the variants, turn those variant catalogs over to scientists, and say, which ones should I care about, which ones could I not care about? And that would also give us insights about how genomic spelling differences play a role in health and disease. So we needed to sequence a lot of people, develop these catalogs, give them to scientists, and have them study it. It's exactly what we've done. The 13 years since the Genome Project ended, there's been a number of projects that have basically tried to get great inventories of common variants in human genomes across the world. The most recent of these, which just concluded, is a project called the 1,000 Genomes Project. This is another big international project where we collected, actually not just 1,000 genomes, we actually collected about 2,500 people's DNA from 26 populations, as you can see, from across the world, sequenced their genomes, developed catalogs, made that available. To give you a flavor of how remarkable that effort has been, is that 13 years ago, we probably knew about tens of thousands of places in our genome that vary among people. Now we know of about 90 million places in our genome that varies among people. We know what those variants are, and that information is all freely available on the internet for scientists to study. And lots of scientists are now studying among those 90 million variants which ones are important for health and disease, which ones are not. That has been remarkably important for the kinds of studies that are going on now. In addition, by sequencing tens of thousands of human genomes, we now have a pretty good idea of what a typical person's genome looks like, which is going to be really important as we start to make this a standard diagnostic tool in ways I'll be describing. Oh, by the way, the 1,000 Genomes Project published their last major paper last fall in this paper. But what about a typical human genome by the numbers? What does it look like? Well, first number to keep in mind is each of us has 6 billion letters, roughly, in our genome. Remember earlier I told you that the human genome can be represented as 3 billion letters, but remember we have 2 human genomes in us, right? We got one from mom, we got one from dad. So when we go to sequence a person, we have to actually figure out the 6 billion letters total that represent your genome. Well, 6 billion, we've now sequenced that across tens of thousands of people. We now know that each of us, on average, contain about 3 to 5 million places, letters, that we differ from one another. So when you sequence a human genome, you've got about a list of about 3 to 5 million variants. Most of those, when we sequence a new genome in a given day, most of those have already been seen. We already have them in databases. That's not new information. But every time we sequence a new person, we get about 150,000 variants that are not in databases yet. Those are the rare variants. They're not the frequent ones. They represent the minority of all the variants you have in your genome, but they are some rare ones that may be just your immediate family or your extended family. By the way, every time we sequence a person, we typically see about 60 new variants that don't exist in either parent. How did you get that? Well, in the process of copying the DNA that went into the germ cells that created you, that machinery for copying the DNA is pretty good, but it's not perfect. And every once in a while, there's a little oops that goes in there that doesn't get fixed. And there's about 60 oopses in you, on average. Probably most of those are completely innocent and don't really have any consequence for your health or disease. But occasionally, that's why you occasionally have a child with a genetic disease that there was no evidence of that in the family because it was a brand new mutation, as we'd say, a brand new spelling that became the very first existence in that particular patient. So that's given us some ideas. It's going to be very important as we start to turn this into a diagnostic test as well. But just because we can sequence somebody's genome and just because we can develop a list of the variants, we still are a long way from understanding whether those variants are important or not. And part of the reason for that is that we don't totally know how the human genome works. I would contend, though, there's been, in 13 years, profound advances in understanding how the human genome actually functions. Now, by the way, you may say, wait a second, didn't the Human Genome Project teach you that? No. Actually, this is what the Human Genome Project gave us. This is only 0.0001%, but this is how you represent the human genome. This is what the Genome Project delivered, the sequence, the A's, G's, T's, and C's in a precise order. It did not deliver an interpretation of that sequence. We always knew that was going to require decades and generations, actually, to fully interpret. We've gotten a really significant progress that has been noted in the last 13 years. We have a long way to go. But what have we learned in 13 years? Well, first thing we've learned is that only about 1.5% of the letters in our genome directly code for proteins. Proteins are sort of the brick and mortar of cells. It's sort of the things that do biological systems and make them operate. Those are genes, protein coding genes. But it's only 1.5%. Now, it's 20,000 genes. We have a pretty good inventory of all of our genes, but it represents only a very small percent of the letters in our genome. We've actually come to learn that there's a much greater percent of our genome that needs to be annotated in a different color, in this case in purple, because it's not a gene, it doesn't code for a protein, but rather it turns out to be very important control units and circuitry that dictate when, where, and how much genes get turned on and off throughout life. And that sort of makes sense, that there'd be some really complicated machinery to do that. Because remember what I told you on the second slide. Every single cell uses the same blueprint. But clearly, a brain cell is going to use that blueprint very differently than a muscle cell. And if somehow they're both using the same blueprint, they must have control elements that determine how to use the blueprint if you're a brain cell, how to use the blueprint if you're a muscle cell, and that's the complicated purple stuff. We've got a lot to learn about how all that circuitry works, but we're getting significant progress in recent years. Oh, and finally, and you may hear about this, there's a whole other language with DNA, which needs a whole other color, in this case green, called epigenomics. Because it turns out that our DNA is not just innocent in being a linear molecule that exists, but rather that linear molecule also gets chemically modified and decorated with different other units in cells. And some of that is called epigenomics, and that also influences how DNA works. You'll often hear about that may be one of the ways that the environment influences our genetics by basically modifying our DNA in ways that change the way the genome functions. So this is incredibly complicated, and it's going to take us many years to figure out. I sometimes say to people that when the Genome Project ended, we knew about that much about how the human genome works. Today, we probably know about that much about how the human genome, ultimately, of course, we need to know that much. This is going to be the thing like a great novel will be interpreted and reinterpreted for generations to come, because somehow in those 3 billion letters or the 6 billion in a person was all the choreography needed for operating a human, and we know there's a lot of complexity in ways that we don't even understand yet. Significant progress, but this is a very fertile area of research as we continue to understand how the human genome actually operates. Well, this is actually very nice. We can sequence human genomes. We know that there's variants that all of us have, and some of those are going to be important in health and disease. And we actually are learning increasingly when those variants fall in functionally important sequences, how they may influence the function of the genome and how that might influence our health, maybe giving susceptibility to disease, which has set up a circumstance whereby we could really harness all these technologies information to better understand how is it that our genomes influence our health. And there has been remarkable advances, I think, in unraveling the genomic basis of human disease. Now, to tell you sort of what has happened, at least give you a summary, let me remind you, essentially every disease, if not having an overt genomic cause, has at least a genomic influence. There's almost not a disease you can name that I won't find some scientific evidence that genomics is influencing at least the severity of the disease. But diseases are very different. You can sort of categorize them into two main buckets. It's a slight oversimplification, but it actually works for describing this. There's sort of rare diseases, and then there's common diseases. And each of them are different in terms of their underlying genomic architecture. So let me explain this. On the one hand, you have diseases that are rare in the population. These are diseases like cystic fibrosis, Huntington's disease, sickle cell anemia. They are rare in the population, but they are relatively simple at a genomic level, because at a genomic level, it is basically a spelling difference, a mutation that is in one gene that basically breaks that gene. And that is the dominant, that's the major reason why you get the disease. You get the mutation, you get the disease. Yes, there might be other variants that exist in the genome that might influence the severity of the disease and maybe even some environmental influence, but by and large, these are simple. They're also called monogenic, one gene. Sometimes they're called Mendelian disorders after the famous geneticist Gregory Mendel. We have made remarkable progress in understanding the genomic basis of rare diseases facilitated by the field of genomics. I'll give you a simple number statistic to keep in mind. 25 plus years ago, the Human Genome Project began October 1, 1990. On that day, at that moment in history, there were 61 rare diseases that we knew what was broken in the genome. We knew the gene that has mutated. 61. Fast forward to today, that number is over 4,300. We've gone from 61 to 4,300. Now, that's remarkable, and we think we should celebrate the fact that there's that much progress, and there's practically every week you hear of another discovery figuring out the broken gene in a rare disease. Still turns out there's about another 3,000 rare diseases we don't yet know, but we're working on that, and there's major programs we have to accelerate that. So that's been an incredible success. On the other hand, rare diseases are devastating to families and to patients, but in aggregate, they don't represent the bulk of healthcare burden worldwide. Really, the main healthcare burden worldwide are represented by a collection of diseases that are referred to as common diseases. Common diseases, like their name implies, are common in the population. And common diseases are ones that turn out to be actually complicated. They are complicated because it's not a single gene, it's not a single spelling difference, it's not a single mutation. It is often, as is shown here, a constellation of multiple spelling differences that influence the severity or the risk for the disease, with what is often a greater contribution of the environment. And yet these are so important because these are diseases like diabetes and hypertension and Alzheimer's and autism and cardiovascular disease and mental illness, and the list goes on and on and on. It's all the diseases you are familiar with, and they're incredibly important clinically, but they're incredibly complicated at a genomic level. We are making progress. One of the ways we need to make progress is by doing very large studies, by collecting thousands of people with each of these diseases, by sequencing their genome, inventorying all the variants, and then trying to figure out which ones are statistically significant and are influencing the disease, which ones are not. When they do influence the disease, how is it that they do that? What are the biological pathways that they're influencing? And needless to say, there's been some incredible success stories, but it is the tip of the iceberg. I think the next 10 years, as we harness this ability to sequence, very many individuals, tens of thousands with different diseases, we will start to appreciate much greater exactly which variants are relevant for these common diseases. So I think we are just beginning to see progress in this arena. It's the area that's so important for us to see, and I think we will see remarkable advances in the coming decade. But in terms of my highlights, notice I've gotten through studying human disease. I've said nothing about the practice of medicine. And to be really candid with you, 6 years ago when I became director of the Institute, I think I probably would have had to stop my talk about here, because I'm not sure at that point in time there were real examples where genomic medicine was truly being practiced. That has changed. I think I can now share with you some vivid examples where genomic medicine is in action and truly are illustrative of what we're going to see in the coming decade or 2 or 3. In reality, I think, and I'm very proud of this on behalf of the field, I think we're beginning to bring genomic medicine into focus. Again, it's still just the tip of the iceberg, but I think the examples I'm about to share with you will illustrate the kinds of early wins, the lowest hanging fruit out there with respect to genomic applications to medicine. So let me tell you about some hot areas. I'll tell you about 5 such hot areas. It's the ones we're most excited about. In many cases, we're supporting major studies, or in many cases, we're seeing others support them because they really are here and now and recognized as being very important. First one is a word I haven't even mentioned, and yet really is the lowest hanging fruit in genomics, and that is cancer. Cancer genomics. Keep in mind that cancer is a disease of the genome. The reason a cancer forms, the reason a tumor forms, is because its genome has picked up mutations, spelling differences that make those cells grow out of control into a tumor. And just as we can open up a normal cell and read out the genome and catalog the variants, we can open up a tumor cell, read out its genome, catalog the differences, and learn a lot about that specific tumor in that specific patient. And what that is doing is really, I think, in many ways, transforming approaches to cancer care. And so there have been many studies that have developed, again, big international efforts to catalog all the genomic changes that tend to be seen in dozens and dozens of different kinds of cancer, and it is giving us clues about better ways to diagnose and to treat cancer. And I will just tell you, especially someone who thinks about some of the diagnostic implications of this, it is beginning to change how cancer is diagnosed and how it is sort of handled by practicing pathologists and practicing oncologists. For decades, right, the pathologists would look under a microscope, and based on what they saw of that tumor, they would make their best guess about what's the kind of tumor, what's the prognosis, what's the best treatment option, and so forth. That's not going to go away. We still need pathologists to do that, but they will be augmented by getting a sequence of that tumor and using the information of the fingerprint, which is shown on the bottom right, of the changes that are in that particular tumor, what that means uniquely for that patient. So morphology will still be an important part of cancer diagnostics, and it will be augmented by genomic signatures that increasingly will teach us a lot about the best way to treat that patient, that tumor, at that particular point in time. This is absolutely standard of care for a handful of cancers now. I think that list will absolutely grow in the coming years. Oh, and if you don't believe me, just go to the web and just see. If you go to facilities that are well known for treating cancer, they are increasingly putting in their advertising material information about genomic testing, and genomics is here and now, and DNA is a centerpiece of what they do, because that is the cutting edge of cancer care, and you will see this increasingly being the case in the coming years. So that's the first highlight, hot area. Second hot area is a big word, pharmacogenomics. Just break it down, pharmacology and genomics, giving out medications and genomics. Well, what does that mean? Well, what it basically means is something we all know, right? Everybody responds differently, right? We all respond differently, right? I'm the guy in the red shirt. Some of your grandkids are probably the kids in the white shirt, right? We're all different. And it also turns out not only are we different how we respond to things like roller coasters, we're actually very different in how we respond to this stuff, right? Medications. Every one of those medications sold at your local pharmacy, they all work. Otherwise, they couldn't sell it. They just don't work in everyone. We are very imprecise as a medical field in how we give out medications. Last year, Nature wrote this review article talking about how imprecise medicine really is, and they listed here 10 very commonly prescribed medications. I wouldn't be surprised if some of you are familiar with some of these medications. And shown in blue are the proportion of people where the medication works. Shown in red are the proportion of people where that medication doesn't work. And yet, when practicing physicians just try to give out a medication, they don't know if you're going to be a good responder or a bad responder, but we're getting better about it because we are learning that much of the reason why people respond differently to medications has to do with genomic variants that influence the pathways that deal with medications in our body. And that some people are better or worse responders to a medication because of variants that are influencing the metabolism of that drug. And the idea is, could we get to a point where we use genomics to help guide not only which medications people get, but the dosage of those medications and so forth? And this notion of taking people in the center there that have the same diagnosis, doing genomic testing of them up front, and figuring out who's going to be a bad responder, who's going to be a good responder, who's going to have toxic effects, and so forth. This is not science fiction. This is here and now. The Food and Drug Administration currently requires over 100 medications prescribed in the United States to carry a label basically saying that genomic information might be useful for making decisions about these medications. And as we get better and better knowledge of variants that are influencing the different pathways responsible for other medications, that list will grow. Absolutely here and now, and you will see this, I think, increasingly grow. Don't be surprised in the coming years if before they give out a medication to you, they will first ask for a genomic test to be done to decide what's the best medication for you. Third hot area, here and now, absolutely, has to do with using genome sequencing to diagnose rare genetic conditions in patients. This has been a remarkable advance because it's come on the scene very, very quickly. The idea is you have somebody, you can't figure out what is wrong with them, and maybe you're guessing and you're thinking about what genes might be affected. In the old days, we'd sequence one gene at a time to figure it out. It's just getting cheap enough and rational enough, just sequence their genome, see what you can learn. In some cases, this has been game-changing. Sort of one of the prototypic examples was from 2011, not that many, 5 years ago. And this child from Milwaukee sort of became the first example of a child whose life was saved because they sequenced this child's genome, having no idea what was wrong with him, figured out what was wrong for the first time, and came up with a treatment plan that has kept that child alive. That was followed the next year, or actually that year, by a similar story with these twins shown here, who I've met, the Berry twins, whereby medical system was simply unable to figure out exactly the right medication to give them for their debilitating neurological disorder, sequenced their genomes, figured it out, recognized exactly what needed to be done. These children are now in college and are thriving, and previously they were clinically going downhill quite quickly. Remarkable stories in 2011, so that by 2012, in fact, Nature predicted that sequencing was going to alter the clinical landscape. It actually absolutely has. The notion now, when disease strikes from nowhere, it just makes sense to sequence their genome and have this be part of standard care. There are now hundreds of patients whose genomes get sequenced now as part of routine diagnostics because they have a rare disease and clinicians can't figure out exactly what is wrong with them. That also has led to the notion of a whole suite of patients that are out there in this world, in some cases who have carried a lack of a diagnosis for decades because the medical system is simply unable to figure out what is wrong with them. And the notion of undiagnosed conditions, in many cases after patients have gone from hospital to hospital, really has come to the fore here in the United States, shown here as a clinical team at the NIH that started a program called the Undiagnosed Diseases Program, which now every single week brings in patients actually from around the world who have undiagnosed conditions. And not only do they get a big clinical workup, a centerpiece of their evaluation includes genome sequencing. And this has been a remarkably successful program. Not that every patient can you figure it out, but in a reasonable fraction of patients, you do figure out what is wrong. Some cases you can come up with treatments. In many cases, you discover new diseases. And it's been proving to be a very effective way to use genome sequencing as part of clinical care. In fact, this program has been so successful, it's being nationalized to multiple sites in the United States, and it's also being set up in multiple countries around the world. So again, hot area, absolutely here and now. Another very hot area actually has two stories associated with it, Genomics of Pregnancy. Here, there actually are two stories because there's sort of two stages of pregnancy. It's before the baby's born and after the baby's born. So let me tell you about each, because they actually have slightly different elements associated with it. It turns out that many people, as you're probably familiar, are very interested in getting genetic or genomic testing of their unborn child. For various reasons, especially to sort of screen for unborn children with devastating genetic disorders. And for many, many years, actually for decades, the only way to access fetal DNA for testing was through an invasive procedure, like amniocentesis or chorionic villus sampling, which is both invasive to the mother, but it also risks the viability of that pregnancy. The world is changing in a remarkable way, because it now turns out that these ultra-sensitive methods for sequencing DNA actually allow you to analyze fetal DNA without doing invasive procedures on mom. Because it actually turns out that an unborn child actually sheds through its placenta small amounts of fetal DNA that then float around in the maternal bloodstream. And we now have the ability to simply do a convenient blood draw from a pregnant mom, and pregnant moms get blood draws all the time, and then subject that blood to the kinds of DNA sequencing that is now routine and so sensitive that you can read out enough information about small amounts of fetal DNA floating around in maternal blood to be able to screen for the same things you used to screen for through non-invasive acquisition of fetal DNA. This is like way cool, and it's absolutely probably the most exciting and applicable genomic medicine tool that's out there, and it's happening all the time. So in a study published last year showed the uptake of this non-invasive prenatal genome sequencing that is done. And you can see by 2014, it was over 800,000 such tests performed worldwide. And I'm told that in 2015, that number surpassed 1 million. So more than 1 million pregnant women around the world are basically getting a new kind of genomic test that didn't exist 5, 6, 7 years ago, all because of the new technologies that have become available for sequencing DNA. And it is replacing, in many ways, as a screening method, these more invasive methods. Really remarkable. That's prenatally. What about the other side of pregnancy? You have a newborn baby. Well, it's interesting. Some news magazines like Time predicted that by 2025, everyone was going to get their DNA mapped at birth. They actually meant sequenced at birth. That led some big question marks, actually, about that. And one makes you wonder whether or not that's a good idea. And immediately, and I'm sure we will talk about this in the session, a lot of questions about should every newborn be sequenced and have that sequence available for their medical care throughout their life. One could imagine some benefits. One could imagine some logistical challenges. You could also imagine some ethical challenges as well. So actually, when Alan Guttmacher, sitting in the front row, was director of the Child Health Institute, he and I got our institutes together and we launched a research program to study this. And that program is now quite mature and is doing some remarkably impressive studies to study the logistics, the ethics, the sort of the applications of should we be sequencing newborns. And we'll see what those results look like in the coming years. I think it will influence what we decide to do. Some of those studies are looking at healthy newborns. Some of those studies are looking at acutely ill newborns. And there, I think there's been remarkable progress, and actually very much is changing medical practice. Because one of the studies in particular funded a researcher to study the applicability of genome sequencing for acutely ill newborns in neonatal intensive care units. And here, once again, you're faced with a situation. Oftentimes, the neonatologists say, I don't know what's wrong with this baby. This baby is going to die probably within a few days. Their hands are up. They don't know what to do. And what they can do now in about 36 hours is to sequence the genome and in a non-trivial fraction of time can figure out what's wrong with the child, in some cases saving them. So what's happening in the United States is you're going to hear more and more about neonatal intensive care units using rapid genome sequencing as a way to try to help them figure out what is wrong with children that are not doing well or newborn babies, whereby it seems if they don't do something quick, they're likely going to lose the life of that child. We're studying this in greater detail, but I'm already hearing this is being used more and more by neonatal intensive care units. Last hot area is hot not because it is successful yet. It is hot because I want to give some realism to what I'm describing to you. And I'm very enthusiastic and I'm very optimistic, but I'll also tell you there's some huge challenges that lie ahead for us. And one of them is just getting this tsunami of information about genomics in the hands of practicing healthcare professionals in a fashion that allows them to do their work. The truth is, as I hope I've convinced you, generating a genome sequence is almost trivial. It's really not hard to do. The problem is we can do that on any patient, but the truth of the matter is today, when we go to round on a typical patient having sequenced their genome, this is what we feel like in the morning. We really don't know what most of those letter differences mean, and we are far from having this convenient to be used by practicing healthcare professionals in that rapid encounter they have with a patient. So we need new systems that allow us to do this. These systems need to integrate with electronic health records and get the genomic data in there. But more importantly, we need that busy healthcare professional be able to pull out their iPhone or their iPad, quickly look up my patient has these variants, what do I do about it? We are so far from having that, it's not even funny. We don't even know how to assimilate all the knowledge that the scientific literature is accumulating, let alone putting it into precise practice recommendations. And so we're starting major programs to try to develop that knowledge base and those routines. And it represents a big challenge that we will all be facing in the coming years, one that we are investing in to try to facilitate. So I've now given you a pretty good romp of the last quarter century of genomics. But I want to transition in the last few minutes and tell you about a few more things. And that is that, and to set this up, I want you to recognize how much this field has changed in a quarter century that I've been involved in it. When I first got involved in genomics, it was just about a bunch of geeky biomedical researchers like me, right? There were people at the bench, we were at the computer, we were just researchers, right? 13 years ago, though, when the Genome Project ended, it expanded. It really became one where healthcare professionals clearly recognized the relevance of genomics, and they came into our tent and very much are now part of this research enterprise to figure out how to use genomics for medical care. But those hot areas in genomic medicine that I told you about, they're starting to be used on cancer patients, on patients who are getting medications, on patients with rare diseases, on pregnant women and so forth, which means genomics is becoming relevant to patients. And once they become relevant to patients, it means it's relevant to all of us, because all of us, if we're not patients ourselves, our friends and our relatives are patients, which means we're part of this conversation. And so genomics is no longer just about science and scientific community, it's about society, because it is infusing its way into the medical ecosystem. This comes with some huge responsibilities, some incredibly complex circumstances, some of which we're studying, some of which we're dealing with. I suspect during the question and answer session, we will talk about how complicated the healthcare delivery system is. You're learning about it in this series. And here we're trying to change medical practice through genomics. That gets really complicated, and indeed, we're facing some of the complexities. We have to deal with issues around genomic literacy. Do patients understand the words of genomics? Do healthcare professionals understand the language of genomics? Remember, my medical classmates, we never heard the word genomics. They're all practicing. Oh, and of course, we also have to regulate this. And there's a lot of issues we deal with in making sure patients are safe as genomics finds its way into clinical practice. These are all major elements of what we are dealing with. They're really hard and they're really complicated, but we embrace them because that's what genomicists do. I like this quote from Winston Churchill, where he talks about how a pessimist sees the difficulty in every opportunity. You could say, wow, changing medical practice, too difficult, we can't deal with it. No, not genomicists. We're optimists. We see this as something that's really exciting. It's an opportunity we have to face. I'm sure that difficulty throws up barriers, but we're going to deal with it. I think it's in that spirit that I really want to just end in the last 5 or 10 minutes and share with you, and I might go a little over time, but I think you'll want to hear about this, about what's happening in the future. Because as this gets infused in society, this gets caught a lot of attention by people who are really quite interested in seeing this go faster and seeing the benefits that I've sort of laid out. And one of those people that's actually gotten quite interested in this is this guy. I assume you know who this guy is. He's my boss, or my boss's boss's boss. But he's actually always really been very interested in genomics. Even when he was a senator, he was introducing legislation into the US Senate, trying to enable advances in genomics. And when he became president, he increasingly got very interested in genomics. And what the story as it's unfolded, and I want to tell you about this because it's something that you'll be able to follow, was that about 2 years ago, June of 2014, he convened a small group of people in the Oval Office and said, I got 2 more years of my presidency. I'm very interested in facilitating advances in genomics, maybe launching a project before the end of my presidency. What should we do in this? And the people that were assembled at that meeting convinced him not to limit this discussion around genomics, because yes, there have been significant advances in genomics because technologies for genome analysis had advanced so much, but rather broaden that view a little bit. Recognize that there's another part of the disease equation that deals with other technologies and the ability to monitor what we eat, our lifestyle, environmental exposures, and so forth, and those technologies are coming. And so the president was convinced to embrace this not around genomics alone, but around precision medicine as a phrase, recognizing that it's not just the G's, A's, T's, and C's that are relevant for health, but other aspects of our life. He obviously was familiar with the idea that if we could account for individual variability, it might present a better opportunity to treat patients in the future. He knew genomics, and he had been kept updated and recognized what was happening, but we really convinced him that there was a lot of new opportunities in monitoring people's physiology as another component of disease, but also their lifestyle, exercise, sleep, also environmental exposures, and then things that sort of are a combination of lifestyle and environmental exposures, like what we eat or what damaging things we do to our bodies. And so the president said, well, what can we do in this arena, and how can we change the practice of medicine? Because he recognized, as I've alluded to, that today too much of medical care is based on what the average patient responds to. But wow, if he could enable through a major program, one that he could get funding from Congress, enable a future whereby we have better ways of measuring variability among people with respect to genomics and environment and lifestyle, that might be a new way of practicing medicine. And he basically said, what can I do to get us from today to tomorrow? So 2 summers ago, many of us were involved at NIH planning something to bring back to the president to have him be able to maybe introduce before the end of his presidency. That plan was brought back to him in the Oval Office at this meeting, actually from about October of 2014. There are a lot of important people in this meeting. The one sitting in the back is actually John Podesta, who, if you don't know, he was a senior advisor to the president, is now running Hillary Clinton's campaign. So this was not just about science, this was also about politics and about policy and a lot of things that would enable this to go forward. The president really liked this idea, and throughout the fall of 2014, asked for it to be fleshed out in greater detail and built in the right budget process to be able to launch it. And then he finally decided he would announce it in his State of the Union address in January 2015. And this is what he had to say.
21st century businesses will rely on American science and technology, research and development. I want the country that eliminated polio and mapped the human genome to lead a new era of medicine, one that delivers the right treatment at the right time. In some patients with cystic fibrosis, this approach has reversed a disease once thought unstoppable. So tonight I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give all of us access to the personalized information we need to keep ourselves and our families healthier. We can do this.
So 3 things you should take from that little clip. First of all, as somebody who's been involved in genomics and the Genome Project from the beginning, no matter it was whether you're a Democrat or Republican, doesn't matter. When the President of the United States talks about your science, that's like way cool. So that was really fun to listen to. Second thing we learned from this was that he named the project. We didn't know what the name of it was going to be till then. It's called the Precision Medicine Initiative. He's the boss, so that's the project's name. Third thing, and I watched the whole State of the Union address, third thing, it was the only time all night Biden and Boehner stood up and clapped at the same time. So this signaled what the president suspected, that he'd get bipartisan support. Indeed, he did get bipartisan support for this. A week later, he formally announced this in a ceremony at the White House, a picture of which is shown here. And off we went, trying to now implement this. There are major elements of this, but the main flagship component of it, which I just want to briefly describe, is the idea of creating a national research cohort here in the United States. Similar efforts are going on in other countries. The US is a little behind, which is why we need to catch up. And the idea is to recruit 1 million or more volunteers in the United States to participate and to be studied probably for multiple decades. It's a longitudinal study. When you agree to participate, you will agree to share your genomic data, your lifestyle information, biological samples, all linked to your electronic health records for scientists to then analyze and mine for years to come. Obviously, we need to share the data, but we also need to build in appropriate privacy protections to make sure that this data is not used inappropriately. And so this is going to happen. It's going to happen, and it's going to provide a platform for studies that are going to allow scientists in unprecedented ways to look at health outcomes, to figure out ways to fuel the development of new treatments, and to catalyze a new era of data-driven and more precise preventative care and medical treatment. Why now? I mean, this idea is not totally new, but it was really recognized that, like the Human Genome Project 25 years ago, was a moment in time to do something historic and audacious. It was to basically go forward on some of these promises in genomics that I have told you about and that really are prime. It also recognizes that now is a great time to harness the data that's in electronic health records. It didn't exist 10 years ago. 10 years ago, only about 20% of healthcare delivery systems in America had electronic health record systems. Now that's 95%, and most of that data is sitting there unanalyzed. It is just remarkably fertile data for analyzing, and we can take advantage of that with 1 million or more people we enroll in this. We also want to take advantage of technologies, new mobile sensors that are going to monitor physiology, environmental exposures, and so forth in ways that previously were not even contemplated. And I think the surge we've seen in technology around genomics is going to be mirrored by what we're going to see in these mobile health devices in the coming decade. And all these devices are going to be talking to these things. These things are going to be part of our medical care, going to have medical information. They're going to transmit information from these sensors into databases. Smartphones are going to be the rage, and they'll be a centerpiece of what's going to happen in this study. You know, when 10 years ago, we only had about 1 million people in America had smartphones. Now over 160 million carry smartphones. And there's going to be a lot of data, and data is going to be a key part of this, and that's so much of what we do, and I think we're ready for that in this data-driven era. And the way the project is being set up is going to basically have people who are participating in this be partners in this. They're going to help plan it. They're going to help control how the data is used. And I think you're going to see sort of social media used in ways that have just not been used in biomedical research previously. So it's a confluence of these areas that have really made this very exciting and a very real opportunity. If you want to read more about it, I'm giving you the most superficial skim of this project, but a lot of information is available on this landing page at NIH. If we're successful, this will be the URL for the next 20, 30 years, because this project will build over time and only get better, and this will be the place where we will put all the information. It is very exciting. We are rushing as quick as we can at NIH to stand this up. The president wants this launched before he leaves office, and those days are getting lower and lower by the week. So we're sprinting to the launch. Some of you might have seen, if you were reading the newspaper 2 weeks ago, lots of activities as we announced a number of major grants to sort of get this project going and get this cohort to start to be built. And even the president got in the scene. He loves all of this, and he's following it very closely. And the day we made our announcement, he wrote this perspective opinion piece in the Boston Globe, talking about how exciting this really is and all these great opportunities. So in closing, in some ways, I've gone full circle, because I started with the Human Genome Project, and now I'm ending with the Precision Medicine Initiative. And I suspect you're going to ask me questions when we get to the question and answer session, maybe about the Precision Medicine Initiative, and my answer is going to be, we don't know. And you may be surprised by that. You may say, wait a second, the president just announced this, how could you not know? I'm actually very comfortable saying that. And it reminds me of what happened 25 years ago. 25 years ago, we were in the first year of standing up the Human Genome Project. If you would have found me back then and asked me, how exactly are you going to map and sequence the human genome? My answer would have been, I don't know. I have some ideas on how we're going to do it for the next year or two, but I don't know ultimately how we're going to make all this happen. That's the nature of this kind of big science, is put out audacious goals, get really smart people together working at it, and figure it out as you go, and that's okay. Don't wait till you figure it all out, then you're starting too late. I think the same thing is true 25 years later as we stand up the Precision Medicine Initiative. There's a lot of things we don't know about this. It just seems like the right thing to do. We'll get all the right people together, and we'll be willing to change our plan as we go along. It worked brilliantly during the Human Genome Project. I think it'll work successful in a similar way with the Precision Medicine Initiative. Because at the end of the day, these are research efforts. We don't precisely know what we're doing. We're trying to learn as we go. And so literally above my door in my office, I have this sign, this quote. I really do believe it. If we knew what we were doing, it wouldn't be called research. So don't be surprised if some of my answers might be we don't yet know. We are researching this. This is what we do. We figure it out as we go along, and that's the thriving aspects of biomedical research. So with that, I will stop. Thank you for your attention. I look forward to the discussion.
And now, without further ado, let's get on with, hopefully, a very stimulating Q&A.
Sorry to take so long, but there are, Ian Sim is back there looking through more of your wonderful questions. It just was an overwhelming task to come up with questions to ask. So I'm going to start with one that was a representative of many, and that is, how can we volunteer to be part of the 1 million people in the Precision Medicare study?
So it's a great question, and remember I promised you I was going to often give an answer I don't know. I won't quite give that answer. What I would say is those sorts of details are just being figured out now. There's going to be several ways to get recruited in. Some of it's going to come through major healthcare networks that are getting grants to be able to get individuals as part of their network into this. But there's also going to be ways to have what are called direct volunteers, people who just say, I want to be in. Inevitably going to be a website that's going to be set up that will be unveiled. There's going to be sort of a major campaign to make people aware of this, so just keep your eyes and ears open. I'm sure you'll be hearing about this. But there will be ways for people to sign up as direct volunteers through a mechanism that will be announced in the coming months.
Terrific. There were several questions on the commercial aspects of reading DNA. What role does the private sector, for example, companies such as 23andMe, play, good or bad, in offering genome sequencing to the public?
So it's a great question. I should sort of set the context a little bit by pointing out that almost from the very beginning of the field of genomics, there has been a considerable interest by the private sector in almost everything we're doing. Sometimes that gets portrayed as sort of a threat. Even during the Genome Project, there was a private company trying to sequence human genome. The fact of the matter is, I actually think most of this has been really good. We must be doing something right if the private sector is interested in doing some similar things. So we've always seen this bubble along, and as I said during my talk, I think it's been fantastic to have the private sector get very interested in technology development around DNA sequencing. If it wasn't for them, we wouldn't be where we are now. So entered the scene in recent years the idea of, well, if you can get genomic information about an individual as part of their medical care, can you offer it to them and not even have a health professional involved? And this often gets described as direct-to-consumer genetic or genomic testing. And it is a little controversial because there is some concern about whether or not people should have that information without having a professional guide them in what that information means. And so one of the prototypic companies in this space is 23andMe. If you don't know about 23andMe, I've noticed there's a lot of TV ads about 23andMe these days. For full disclosure, 23andMe, I know them extremely well. In fact, my institute gives grants to 23andMe to do studies that we're interested in having them do with the data they've collected on individuals. One of the things I think that's been really good about 23andMe is that it has created a lot of interest in genomic medicine and information about your genome and how it may relate to your health and possible disease and so forth. So I think it's stimulated a discussion, which I think has been very valuable. Where it has run into a little bit of issue is around the question of, are they overreaching? Are they surging ahead of what we currently know, what a meaning is of certain genomic variants that we have, spelling differences, and what that really means medically? I'm not the only one who is, and others in the community have gotten a little concerned about this. I'm just telling you known facts. The FDA got very concerned about this. And in fact, the FDA stepped in maybe a year and a half ago, and actually said they had to stop giving medical information unless they sort of pass certain thresholds, which they've actually now done, and there's certain things they can do now, and there's certain things they still can't do. One of the things that is people I know are particularly interested in is a lot about ancestry. People have gotten very interested beyond the medical stuff, just about having your DNA tested either with 23andMe or Ancestry.com and other companies. And so, it's one of these fine balances. They are excited about trying to enable people to own more of their own medical information and ability to get information about themselves, but we want to make sure it doesn't go too far and have people. What I worry about is that people will overinterpret and find out that some of it was a little premature, and then they'll say, this genomic medicine stuff, it's not real, it's not, and that was just because maybe they overreached. And so it's a fine line we're drawing, but I think by and large, my conclusion is I think this has been healthy for the ecosystem that I work in, and I think, although we just need to sort of have checks and balances on it.
I know there are a bunch of medical personnel in here, and this question is, how will genomics affect the clinical course of training new physicians, healthcare professionals, and the public?
It's something we think a lot about, and we're not alone in thinking about this. But there's multiple facets, and I just briefly showed one slide to sort of whet people's appetite about that. Because first of all, we have to deal with the next generation, right? So we have a third-year medical student friend of mine sitting in the front row. How are we going to train her to deal with this completely new world we live in? In some ways, that's a little easier because she's a captive audience, and she has a test to study for. Or we could say you got to learn this, but we still need to update medical curriculum to deal with a whole new way of dealing with this. That's easy. What about my medical classmates? My medical classmates, we're all in our mid-50s, and they're going to practice probably another 1 or 2 or 3 decades, maybe, and they never heard genomics once. And if they're an oncologist, they're completely changing what they do. If they're an obstetrician, they're completely changing things of dealing with prenatal. If they're a pediatrician, they have a whole new world of genome sequencing, and yet they're busy. And they're overwhelmed, and the medical ecosystem is not a very friendly one these days for them to work in. And somehow we need to get them updated. And so that's probably even the bigger challenge, is how do you deal with, and yet, this is a large swath of them. And by the way, it's not just physicians, I should immediately point out. Of course, it's nurses, and it's physician's assistants, it's pharmacists, it's, genetic counselors. So we need to deal with, so there's lots of things happening. We work a lot with professional societies. We deal with organizations that try to help with this. I think we need something bigger and bolder, and there's some things we've been thinking about and strategizing about how to really deal in a much larger scale with genomic literacy among professionals. And let's not forget about genomic literacy among the general public, because they're going to need to speak this language with their healthcare professionals, and they need to understand basics.
I know that as a lawyer, we had requirements to have continuing legal education if we were going to keep our licenses. I assume the same
Continuing
is
medical
true.
education, sure, there is, and that could be built in to some extent as well, and those are some of the efforts. Probably even need to be bigger and bolder if this continues to happen as fast as it seems to be happening.
Question here about what is the effect on life insurance going to be with all this additional information?
Complicated. One of many complicated societal issues around this, and and we could unpack many other. Life insurance is one component, long-term disability, health, et cetera, et cetera, et cetera. And I think we're going to, for some of these things, some of the work coming out of our institute historically around the ethical, legal, social implications of genomic advances materialized in some legislation being passed by the U.S. Congress, something called the Genetic Information Non-Discrimination Act, which prevents discrimination, but not in all sectors. It doesn't necessarily cover life insurance, for example. So I think we as a society are going to have to grapple with this. What are we willing to do? Should it just be everybody as a society, we should protect each other? We were all dealt some genomic cards. We didn't have a choice of what they were, and we need to somehow reckon with this as much as we reckon with lots of issues around social equality. And these are not going to be simple answers. And I'll also tell you, every country is dealing with this a little different, which is probably not surprising, of how they're sort of embracing or not embracing genomic information as part of what can and cannot be used for various aspects of medical care and employability and so forth.
Thank you. Would you discuss CRISPR and the technology and name some of the ethical issues changing the genes in the genome? And please take a position or two on these ethical issues.
So let's talk about CRISPR, and I assume nobody has dinner plans, right? Because if we're gonna talk ethical issues, we're gonna be here a long time. So what is CRISPR? I'm impressed that there's been, from what I understand, many cards coming in. And I would also immediately tell you I am not an expert in this, but I can explain it in very general terms and say why it's relevant. So think of it this way, is that we have technologies for reading DNA, and wouldn't it be nice if we had technologies for editing DNA? If there's a G when you want a C, is there a way to strategically go in at that specific place and modify it? And there's been various technologies that have been developed, and the really cool, exciting new one is something called CRISPR, which the reason it's called that is geeky and you don't care about. Bottom line, though, this is an incredibly impressive technology that allows you to tactically go in and make very specific editing changes to DNA. So that sounds great. And I'll tell you, as an experimental tool, it's awesome, because you could take a yeast, or you could take a fly, or you could take a worm, or you could take even a mouse, and you could modify the genome at a very particular place, and you can figure out what happens. And you could then use that information to understand human biology, because we used a model system to be able to do this. And so there are unbelievable numbers of very sophisticated research studies that are using this technology to manipulate genomes of model experimental systems. But then there's human, right? And so one could imagine all sorts of possibilities, but one could also imagine all sorts of ethical issues that come up. There's ethical issues around the concept of, do we really know what the implications are of making this change, and that there's no other changes that take place? And in fact, while CRISPR is really impressive, it sometimes makes some other changes here or there. Well, maybe that's okay if it's a fruit fly, but you're not really going to be okay if it's a person. You don't want extra oopses coming in, and so we have to be a little bit cautious about that. So there's a lot of ethical issues of it. When is it time to try to use this? I think then there gets to be issues around about what would be appropriate and inappropriate to do. if we're going to start editing people's genome in terms of what, you know, if it's a devastating disease, it's really simple to change. Okay, that's one thing. But what happens if it's going to have some other aspects that maybe not everybody agrees is either a disease or is some change to an attribute people may not be comfortable with? So there's a lot of issues just around its use. But I also want to temper a little bit to keep in mind the following, is that the only way this is going to be effective deals with tissues or cells in our body that you can actually typically get that are beaten copies, cells that are turning over. And so CRISPR as a means of gene therapy or of gene editing is most likely only going to be helpful for cells that are sort of replicating and still growing. And so there's a limitation for some things, and this has been true of gene therapy actually from the very beginning. So for example, some diseases may only affect a subset of cells in the brain, like Huntington's disease, a very small subset of brain cells, and these are cells that aren't necessarily turning over. So that's a problem, and also accessing brain cells is pretty difficult. Most people don't like you going in there and mucking around with the cells, right? Okay? Whereas other diseases might be caused by cells of your bone marrow, for example, certain blood diseases or certain diseases that affect your immune system. And there, those cells are turning over, you can access bone marrow, that's much more straightforward. And so some of the big successful stories for any kind of gene therapy have been cells that access, that are turning over, that you can grow, you can put back in. But that's not true of a lot of diseases. And so, you know, yes, it's very exciting. Yes, one can imagine all sorts of things, and I'm sure we will see this. But there's still a lot of concerns about how it's going to be used, and also, and especially when you, and this is probably underlying part of the question, is the idea of making these genomic changes in embryos and some of the implications there. And right now here in the US, absolutely not allowed, inappropriate, et cetera, et cetera. There's been rumors of other countries, I won't name the countries, but there are rumors of other countries where there has been some embryo manipulation using CRISPR. And so that's created quite a stir in the scientific community.
And that raises questions about testing and selecting embryos created by in vitro fertilization. And are we heading to high-tech eugenics selecting for superior human beings?
Well, that's easy. Great question. I should preface this by immediately pointing out, like a lot of technological advances, there always seems to be two edges to the sword, right? I mean, it always seems that there's just some incredibly compelling, wonderful things, and then you can think when you use it for evil, it can be just some awful things that totally terrify you. We need to make sure that we enrich and protect people. I mean, enrich for the good things, protect people from the bad things. There are lots of remarkable things that can be done nowadays around with respect to making sure parents have the opportunity to have healthy babies. One of the examples of this, which actually is not even that new of a technology anymore, probably a couple decades old, I suppose, is something called pre-implantation genetic testing. So couples who want to have, they do in vitro fertilization, and when that fertilized cell becomes about a 16-cell stage, you can go in under a microscope and you can remove one cell and test it for a genetic disease. This is often for parents that have already had a child with a devastating genetic disease. They know exactly what they're looking for. They can do a DNA test and figure out whether that embryo is going to have that disease or not. And you typically, when you do in vitro fertilization, you have multiple embryos, test one cell from each one, decide what are the good ones that you might want to implant, one of the ones you don't want to implant. And this has gone on around the world thousands of times. And for devastating genetic diseases, people I think are generally very comfortable with this, and there's many success stories. But you raise the underbelly of it, which is, oh, wait a second, what happens if there's other things we could test for? Let's test male versus female. What happens if we figure something out about some other attribute, a tall child or an athletically enabled child, any of these things that one could imagine coming up with ideas for. You start to go down a gradient where it starts to become more and more and more and more uncomfortable to people in society, and we've got to decide where those lines are drawn. And people won't always agree, and different countries will choose to do different things. But I think for the medically compelling cases, it's just awesome what we've been able to provide as genetic, is to make sure that parents have choices here. But when you start to think about other scenarios, it makes people very uncomfortable, as it should.
Can you hear in the back?
To me? For me? Oh,
Yeah.
wow. people usually always hear me. Okay, easy.
So Eric, are
Yeah.
there organizations who are trying to control the use of these techniques and have an influence on the ethical use of?
I think the professional societies certainly come to mind. I mean, first of all, we pay very careful attention to this. We formally study this. Actually, Congress has asked us to do it. We are the only, I think we're the only institute that carries congressional language in our what's called our authorization, the way we have to operate per Congress, that we are required to dedicate 5% of our research dollars to studying the ethical, legal, and social implications of genomic advances. So many of the things we're talking about, which are these ethical issues, we're studying them. We're giving scientists and bioethicists and legal scholars, so forth, grants to study this, to help inform policy decisions, to help inform government from making policies, help inform professional societies from taking positions and helping this. There's many aspects of the ecosystem that sort of oversee the practice of medicine. And yes, there are absolutely organizations, both private foundations, professional societies, and so forth. And they help us quite a bit, and they take positions as they should.
Do you encourage regular folks, not patients, to get their genome sequenced?
So it's regular folks, because all of us, who's regular these
Well,
days?
If we're not patients.
Yeah. So, but all of us are patients to some
We are
extent.
at
So the
some
question
point.
is, let me flip the question around, because it's a question someone's going to ask me. I'm sure it's in the deck of cards here somewhere if I had my genome sequenced, right? I mean, here I am. So I've not had my genome sequenced, in part because the vision I lay out for the future is not, it doesn't immediately say that all aspects of medical care require a genome sequence. But there's certain scenarios that do, whether you take certain medications, whether you have cancer, whether you have an unborn child, whether you have a rare disease, and so forth. So I'm very transparent in telling people I have not had my genome sequenced yet. I would in a heartbeat if I thought it was going to be medically relevant. There's two aspects of my life where this was medically relevant. I am of Ashkenazi Jewish descent. And so when my wife and I went to have children, the standard was to make sure I was not a carrier of Tay-Sachs. So I was genetically tested then. I'm not a carrier, so it was not an issue about an unborn child. That was point one. I didn't need a genome sequence for that. If you look at, don't ignore the family history being incredibly important here, right? And so you should know a lot about the health history of your family. That says a lot. It's genetics, right? And so it's saying a lot. If you look at my family history, I am at significant risk for melanoma. I mean, I have coming from both sides of my family. I don't need a genome sequence to guide me on what to do. I know I'm at risk. If I sequence my genome, I might get a clue about why I'm at risk, but it wouldn't change what I should do. I should once a year go see a dermatologist and have him or her check me out head to toe. And I should look for anything that looks suspicious. So I have yet to encounter, and I'm not on any medications, and knock on wood, I'm generally healthy. So I haven't needed a genome sequence yet, so I'm not going to run out and get one. I'd have to pay for it out of pocket anyway, and we could talk about healthcare and at what point they'll reimburse, and that's a big issue right now, because even with some kinds of cancer. If I got cancer tomorrow, I'm not guaranteed. I would absolutely get my cancer's genome sequenced. I'm not guaranteed it would get paid for. I'd probably pay for it out of pocket. I would pay for it out of pocket if I had to. But insurance companies haven't yet willing to pay for that. Hopefully that will change over time. So I don't think ordinary people necessarily should, unless you're curious and you want to learn about it. Going back to 23andMe, I think one of the positive things about 23andMe is when people get it. That's not a genome sequence, by the way. It's only a partial inventory of the variants that you happen to have, not a genome sequence. But it has motivated non-medical, non-scientific folks to learn. They use that information to go in and say, wow, I have this, or I have that, or I'm this, my ancestor. So from a recreational point of view, I think if you're interested in that, sure. But just don't overinterpret it medically. I'm not sure genome sequence would be all that useful for if you're otherwise healthy. But again, from my perspective, if you encounter a medical circumstance where the healthcare professional says, I would be aided by genomic information on you, I would certainly get it in a heartbeat.
Okay, we had a number of questions on specific diseases, and I'm just going to run a few of them by you.
And I'm probably not going to have the expertise, but I'll try.
The question is there research being done, for instance, on ALS?
Yeah, absolutely. So there's practically not. So here's what's happening right now, is that So I run one of the 27 institutes at NIH, but I don't own a disease. I mean, there's not a single disease. You know, we're worried about the genome. We're not worried about any specific disease. We're worried about how our work can help study other diseases. But then you have most of the other institutes fall into major areas that are studying specific diseases. ALS, the Neurology Institute worries about that. Cancer Institute worries about cancer. Mental Health Institute worries about mental illness, so on and so forth, right on down the line. And so what is happening, which we think is fantastic, is that all these other institutes are working with us, and we are helping work with them and teaching them to do the kinds of studies that we think are necessary. And so a lot of partnerships, and they're taking, such as with ALS, or they're taking Alzheimer's, or taking autism, and designing very large studies, thousands and thousands of individuals, sequencing their genomes, analyzing the data, seeing what they can learn. And it's, to us, this is sort of a testimony of the success of genomics, is that we're seeing it spread all across all the other parts of biomedical research, which is exactly what it should be happening.
Well, of great interest to me and many in the audience is what can this research do to help those areas that are simply the result of aging, like arthritis, stiff joints, I'm remembering one of the questions, and it listed all of many of the symptoms that we have. Is there something that this could?
Well, one of the things that is happening, and part of the reason why, this is probably a really important point I should make, if we sort of take these common diseases, whether it's arthritis, or it's lupus, or it's Alzheimer's, or it's cardiovascular, coronary artery disease, or it's asthma, any of these common diseases, one of the things that's coming out of these large genomic studies isn't necessarily to be able to predict whether or not people are going to get these diseases, because that's often going to be very hard to do. You know, these diseases I just named, they have a genetic component, they also have an environmental component. You know, think about, we can tell story after story after story. Why is it some people who smoke never get cancer? Why is it people who never smoke get lung cancer, and so forth? It's not always predictable. I am not sure for common diseases how much genomics is going to be helpful for prediction. Family history probably would be helpful, but it's not, even that's not a guarantee. But the main reason at the moment why we are doing these studies is we're trying to figure out what are the variants in genomes that make you a little more predisposed to get arthritis. Why do we want to know that? We want to know that because we want to understand the biological pathway that has been disrupted to make you get arthritis. Because if we understand that biology, we might be able to either identify a current drug that can be used for that, or maybe design or develop a drug. So much of common disease, we just don't understand the biological pathways that are broken to be able to come up with therapies. And every time we think we know, we realize we're pretty naive. I'll give an example, because I think it's one of the earliest vivid examples coming out of these studies where you're taking lots of people and trying to study genomic variants and figure out what is conferring a risk for disease. Reasonably common disease, age-related macular degeneration. We probably all know people who have this. And scientists have been studying for years. They thought they knew exactly what was, or maybe they had ideas of what was wrong. It turns out when they actually did one of these big genomic studies, they found out that what pathway was disrupted was a complete shock to them. It turned out it was an immune disease. And the pathway would have never been guessed to have been involved in this degenerative eye disease. And it immediately gave ideas about new therapy. And now there's new clinical trials ongoing precisely for this, whereby new therapies which previously had never been envisioned for this disease are being tested. That's the kind of thing we learn. And so, again, for almost any of these chronic diseases, what we really want to understand is the pathophysiology. What are the biological pathways that are disrupted?
Here's a question on ethical issues deciding on who gets organ transplants based upon their genetic or genomic makeup, information, and predictions. Have we progressed to that?
So I'm not very, so I guess from the question it implies that some people are not getting access to organs because they do genomic testing on.
Well, I think the question is, may genomic testing be used
To better match.
Yes.
Yes. So right now, obviously, there's a lot of work that goes into more precisely match organs to individuals, and it will, to my knowledge, but I don't have all this expertise. To my knowledge, I don't think we've advanced to the point of doing genomic analysis to learn the things we need to learn about proper matches. But maybe that is changing. One thing I will point out, which I think is another way cool thing that is relevant to transplantation, is remember when I told you that we can test fetal DNA that floats around in maternal blood? What I didn't mention at that point, because it wasn't the right time to mention it, is that same technology for monitoring free DNA that floats around in our blood can be used for monitoring for cancer that might float around in our blood, and there's many companies that are being set up now to potentially screen people, even healthy people, to see whether there's any evidence of tumor DNA floating around in our blood as a means for screening for cancer, or for people who have cancer, get treated. You always wonder, is it coming back? Is it coming back? You typically get radiographs and so forth. The idea is you can test their blood and test that to see whether the cancer has come back. Another application is people who have a transplanted organ. They are monitoring those people for life. It's an organ from another person. That person has a different DNA composition than them. They do all sorts of monitoring to see if there's any evidence of rejection. There's work that's going on to see whether or not a better way for monitoring for rejection is by monitoring for DNA. You know what DNA to look for from that organ floating around in the bloodstream of that patient. Because if the tissue starts to get rejected, they'll be shedding DNA from cells that are getting broken open and so forth. So another way cool technology application that's relevant to transplantation.
Another question about Alzheimer's, and I am assuming the answer is that the neurological part of NIH.
Actually, the Aging Institute, just related, actually, it was very interesting. The president, once again, coming back to the president, being very interested in genomics, but also recognizing, as he very interested in healthcare, obviously with the Affordable Care Act, recognizing that in the long run, Alzheimer's and related disorders are going to present major challenges to healthcare. We all know that. We can appreciate why. As we live longer and more of us develop these disorders, that it will be a very large burden on the healthcare system, and wanted to sort of see what could happen if we understood the genomics of Alzheimer's. And so early in his presidency, actually about 5 years ago, he stepped in and said, I want a major genomic initiative in Alzheimer's. Once again, he's the boss, so he gets to say what he wants. And so we actually got together. We formed a partnership with the National Institute of Aging, which is the major funder of Alzheimer's research in the United States. And we have a very large study that's actually quite mature now, taking lots of individuals and getting genome sequencing and trying to see what we can learn. Some very interesting things coming out will probably be reported in the scientific literature in the coming year. Again, trying to move quickly to see, are there pathways we've never figured out are relevant in Alzheimer's? And would those give us clues on how to slow down progression of the disease?
I got a bombshell for you. Whoops.
Uh-oh.
Whoops.
Oh, that's not a good one. I can already see that from here. That one's hard.
The question is political.
Oh, politics.
Does the scientific community have a feeling about the upcoming presidential election?
Are the cameras rolling?
You know, it's surprising how many of the people who are answering these questions ask that question.
If the camera's rolling?
Yes.
Well, you ask a federal employee, he really wants to know if the camera's rolling. I can get in big trouble if I enter the fray of partisan politics, especially election. There's a whole law out there, it's something called the Hatch Act. But I'll answer it this way, is that I, just by pure observation, without in any way signaling any favoritism on that, I think so far in the presidential campaign, there has not been much discussion about biomedical research or interest in science from either of the parties. That said, it does seem that once we get into the real election season with two nominated candidates, oftentimes the professional societies will get involved and will get in touch with the campaigns and at least ask for position papers, and oftentimes they do a lot of arm twisting to get things that, if not being talked about, at least being posted on websites. So I certainly hope there'll be some discussion. I think it's an important part of this country, And I do think we should hear from both candidates about their interest and support of and their vision for biomedical research and its place in society and in the government.
I
Was that good?
No, I was good. I was going to ask him if he thought both the candidates understood what biomedical research was, but sorry about that.
Did you notice I'm not biting?
I know. Will genomics provide information that will make the development of new drugs less expensive and/or faster?
Yeah, it's a great question. You know, if we're not talking about the complexity of healthcare, we can start talking about the complexity of drug development. And I will readily admit I am not sophisticated in fully understanding that ecosystem. But I've learned a lot over the years. Here's what I would say. I would say two things. First of all, what I hear from many whose lives are all about in the pharmaceutical industry and the biotech industry is that they simply don't have enough what are called targets, enough good ideas about what things they should be developing drugs for. So I think genomics has a promise, and we'll see if it fulfills the promise, but it has the promise of surging us ahead by doing these very large studies that are going on now that will give ideas about, I need a drug for this, I need a drug for this, this pathway broken, this. Hopefully, we will provide more targets, which would give them a wider set of things to pursue to potentially feed more things into their drug development pipelines. That's the hope. Now, the second thing that's happened that I've had only just a small amount to do with at a very early stage, but I really deserve very little credit for it, and the credit really goes to the current NIH director, Francis Collins, is recognizing that there are certain things about the drug development pipeline that sort of aren't very efficient, but pharma's too busy trying to get it developed that they don't have time to try to improve. And so one of the things that our NIH director has done on his watch is to create a new center at NIH. And that doesn't happen overnight. There's 27 institute centers. It's a big deal. Got to get Congress behind it and all that. And basically to create a center that is trying to come up with technological innovations to improve the pipeline of identifying targets, developing drugs, and so forth. And we hope that these advances will do to sort of drug development what genome sequencing is doing to genomics, is to sort of knock down barriers along the way. So again, I think it's another good example where I think it's recognized this is a problem, and what can the government do unique from what pharmaceutical, and where do they work in partnership? This is one of the ways. This is trying to approach this a little more aggressively, seeing if we can help more. Because if we can help in generic sense, then the private industry is going to be healthier by being able to use those technologies and go quicker. But there's no question that this is a big issue because the cost of developing drugs are huge, and of course, of course the other thing I guess I should throw out, although we don't yet know what this really looks like, is a lot of drugs fail because a certain fraction of people have significant adverse reactions to those. Sometimes those are drugs that never make it to market. We need to be systematically going back and looking at some of those and see whether or not, if we would have had genomic information in advance, whether we could have predicted maybe it's not such a failed drug. Maybe if it was guided by genomic information, can we salvage drugs that previously were thrown away because of a subset of people had really adverse effects. Increasingly now, I think pharmaceutical companies are recognizing that getting genomic information up front might be very helpful for the drug development testing that goes on very late. So that's a to be seen.
This is kind of a little twist on that. What is the role of the FDA in the emerging world of personalized medicines? How can they possibly measure the safety or side effects of such individually tailored medicines which may not have gone through clinical trials?
So the first half, let me try to dissect this because I want to sort of influence the thinking around the second half. Some people think that when they hear about getting genomic information, we are then going to use that information to tailor the development of medicines for each individual person. That's really not what this vision is. What this vision is among the set of medications that have already been approved and looked at, what's the right medication for the right person. Because the idea of developing a drug for one patient is a completely mythical idea because it's way too expensive and way too long lead time. So for most of the applications we're talking about, it's not designer drug development for one patient. It might be designing of drugs for patients who have this pathway broken, but there's probably thousands of patients once that drug gets to market that will benefit from that, tens of thousands hopefully. So again, that's a slight clarification that's really important. In terms of the general oversight of the FDA, this is a three-letter word I almost had nothing to do with for the first 20 years of my career in genomics, in that because it was not touching medical applications. The last 5 years, we are interacting extensively with the Food and Drug Administration, and as I told you earlier, they're getting quite involved in things like 23andMe and other things we're doing. And in fact, even the program that Alan Guttmacher and I launched around newborn sequencing, the FDA jumped in and said, wait a second, you're going to start now sequencing genomes of children. We have to be involved. And actually, when we launched the program, we didn't think the FDA was going to even care about this program. So there's a very deep engagement we now have with them. Their job is to make sure that the proper regulations are in place, that if we start to do anything to change medical practice, patients are safe. And, that's a whole art associated with that, and that relates even to a diagnostic test, and genome sequencing is certainly becoming a diagnostic test. So they're getting very involved in this, and they need to be. And actually, part of the precision medicine initiative that I didn't even talk about relates to having the FDA get much more advanced in how to deal with some of these aspects of precision medicine.
Will congressional budget cuts affect the precision medicine initiative or NIH ability to continue with genomic research?
Yeah, so cuts is a four-letter word I despise. We are completely reliant on the generosity of the U.S. Congress to provide adequate funds for biomedical research in the United States. Cuts are devastating. The sequester that happened a few years ago was devastating. In general, there is insufficient, here I will get up on a soapbox, even if I get in trouble, there's insufficient funds coming to the National Institutes of Health and other biomedical research groups here in the United States. And we have seen in real dollars a decline in support over the last decade at a time when I actually think we should be pushing the accelerator and increasing our support. And I will tell you that other countries are doing a better job of supporting biomedical research than we are. They're taking our playbook, these strategic visions and genomics that I outlined, and they're running with it more aggressively than we are, and I think that's a shame. So it's gotten better of late. This is a good year for NIH. And some of the early signals for next year are positive. But we still haven't crawled out of the hole that was created by the sequester. And all I can tell you is we could go faster. We would go a lot faster if there was more money. Now, there was money provided for the Precision Medicine Initiative, so that's up and going. Assuming that money is not cut, it'll continue. It needs more. And we certainly hope that the next administration, the next Congress, the next everything will hopefully even be more generous, because we need it, and at many levels we need it. And it's not just to keep us employed, it's we genuinely believe we can go faster, and we generally believe we can help people if we have adequate support.
A couple of questions on Joe Biden's moonshot on cancer. You got any thoughts on that?
So, sure. I should immediately point out that we have a very large institute at NIH called the National Cancer Institute. It's 10 times bigger than my institute, so I'll just immediately point out this is a huge, as it should be, a huge enterprise, and pretty much they are owning this, working with the vice president, and there's a number of things that they aim to do. By the way, this is so far mostly an idea and a proposal. It has not been funded yet. This is one of the things that has to happen in the next budget year, is to get funds for it. And the vice president has engaged the scientific community, actually in a way quite similar to the way we have over the years engaged the genomics research community. What's needed? What are the barriers? What are the changes, cultural changes even, about what should happen? And he's even done this in other countries. He was just in Australia, I was reading, and trying to get ideas. One of the things I know he's very interested in, and I am completely supportive of, in something that has really grown up. Remember, I showed that article that Jim Watson, Francis, and I wrote last fall about 25 years of big science. One of the things that genomics sort of changed culturally in biomedical research is the idea of data should be shared. Data should not be hoarded by a scientist and then let them write. Data should be shared openly because inevitably, the more the data gets shared, the more data you have, the more eyes you have looking at it, the more scientists you have analyzing it, the better. And we were game-changing our community when we started sharing Human Genome Project data long before we published it. And the internet facilitated that, and that is now standard. Data sharing is a huge thing of what we do, and increasingly more and more what goes on in biomedical research. The vice president has learned that in some cases, major cancer projects are going on and the data is not openly shared, that it sort of is contained just by the groups that are collecting it, and he wants to do many things to facilitate larger scale sharing of data. And I think that's a great thing because we really need large numbers and large amounts of data and many more people analyzing it to get the kind of advances that are going to be needed.
What is the current legal status of companies filing for patents of genes they discover?
So another area of a societal issue that indeed our institute studied and actually gave a lot of grants out years ago around sort of the intellectual property issues surrounding genes. And that was, I think, very informative for eventually when the Supreme Court took up this famous gene patenting case, which actually went through a couple, didn't get resolved at first. I actually got involved, and other people at the Institute got involved in helping to brief the Solicitor General of the United States, actually two different Solicitor Generals of the United States who are the people that argue the government's case in front of the Supreme Court. So we were very tied into that, and we were delighted when, about 2 years ago, when the Supreme Court ruled against gene patenting. We agreed with the position that ultimately was taken that, sort of this DNA is out there, it's everybody's, and the idea of what was happening, almost setting up toll booths along the DNA where people would have to pay into patents and extended, fees would completely undermine this vision of having genome sequence be a fundamental diagnostic tool for the future of medicine. And so it's, so that's been taken down. You can't patent genes. There are still some nuances around what you can patent, and that'll work its way. But I think it has been a very important ruling by the Supreme Court that we embrace.
How will genome research allow us to look at early humans, and what are we likely to learn about our ancestors?
Great question, because it was an area that didn't really directly relate with medicine, so I didn't touch on it. But again, I keep using the phrase way cool. It's just what I think it is. It's just way cool that one of the things that has materialized from these incredibly sensitive methods for sequencing DNA is that they allow you to sequence ridiculously small, degraded samples of DNA and get information. Now, what does that mean? That means that you can unearth small pieces of our ancestor, like Neanderthal, little pieces of bone, just incredibly small, that have been excavated from some cave that are, millions of years old, and you could very carefully extract a small amount of DNA out of it, and you could actually get good data using these new technologies. And so this is called ancient DNA analysis. It's a whole field of study. I would not be surprised if there's a Nobel Prize given for this in the next 5 years or 10 years. And it's been remarkable. It's teaching us a lot about the history of the human species and especially its migration patterns. And it's given us some clues, and there's jokes now, because now that we know all of us carry a small amount of Neanderthal DNA sequence in us, and some people think they have a little more, a little less, and it immediately gets... It's one of the things you learn by getting 23andMe analysis of you is how much Neanderthal you have in you. And so it's been, it's fascinating. It's fascinating. And you'll read articles about, one of the articles that you probably read recently is they always thought Neanderthals were sort of over here and modern day humans were over here, and they never interbred. And there was a little hanky-panky that went on between those two lineages, because they actually find evidence. Genomics doesn't lie, so they find out exactly what happened. But it teaches us things about the history on this planet that's intriguing. And, again, who would have ever thought that that would ever be possible? But it actually turns out it is. So, similar to how it teaches us in more recent ancestry, I think people who are very interested in genealogy, people are very interested in tracing their ancestry, the power of having DNA analysis behind this is just remarkably strong. And a lot of people get their DNA testing done for ancestry information.
Well, I've saved this one for last because I knew you weren't going to touch on it. Please discuss the implications of the genomic study of microflora, fauna, etc., found in the human gut.
Sure, absolutely. So this is another area I didn't even touch, in part because of time, but is also a reflection. Again, it's maybe the thing I should say is technology just drives everything. I mean, if you sort of look around, and even the history of science, think about what the telescope did for astronomy, thinking about what the microscope did for cell biology, and the DNA sequencer is doing this as well. It's just opening up things we never thought was going to be possible. So the story basically goes as follows, and you're going to get immediately uncomfortable. So your human body is not mostly human cells. The great majority of cells in and on you are microbial cells, not human cells. So you have a little ecosystem in your body, and you're the minority owner of it. You just may not appreciate it. You are greatly outnumbered, about 10 to 1, or even more. You are greatly outnumbered by the number of cells. A lot of them in our gut, but a lot of them are in other places, our mouth, our ears, certainly all over our skin. Don't think for a second your skin is sterile. You got so many bacteria on your skin, you have no idea. So another interesting thing about this is that the great majority of those microbes, I mean like over 90% of them, up until a few years, we didn't even know what they were because we could never culture them in the laboratory. When you go and culture anything in the laboratory, in any standard microbiology lab, only a very small fraction of what's in you and on you will actually grow because we haven't figured out how to get them to grow outside the human body. So we sort of were blind to all these other critters that were in us and on us for a long time until sequencing became cheap and sensitive because all those critters have DNA. All those critters have the genome. They got G's, A's, T's, and C's. And so instead of trying to isolate them, we can just inventory them by sequencing. So you could simply take a cotton swab and swab your skin or take something out of your mouth or your nose or take a little bit of your GI tract and just purify the DNA, sequence it, and you just read on the computer, you can figure out what living forms are there, what bacteria are there. So that's like remarkably interesting because all of a sudden it's like turning on the light and seeing what is the living forms that are in us and on us. By the way, they're living in us and on us. They have genes, they're producing proteins, they're doing things, and we don't think about it a lot, but we probably should. And we actually always had a suspicion that they probably live in a healthy sort of an equilibrium with us most of the time, but some of the times they may actually influence, either cause a disease, or maybe when we get a disease, maybe they sort of change what's happening. And this is, so by the way, the collection of microbes in a given environment is called a microbiome, microbiology, microbiome. And microbiome research is super hot right now, another super hot area. And why? Because of DNA sequencing. So now there are all sorts of studies. There was a big project, actually it had an international flavor called the Human Microbiome Project. Sound familiar? Instead of studying the human genome, study the genomes of all the microbes in us and on us. But also now many studies going on to look at what are the changes in the microbiome associated with health and disease. And it turns out and by the way, the microbes, I like to think of it, the microbes are our most intimate environmental exposure. They're not us, but they're sort of part of our environment, but just happen to read them out by looking at their DNA. And so what's happening is remarkable studies where they're looking at patients who are diabetic, patients who are obese, and looking at their gut microbiome. It turns out there's some very interesting correlations. Looking at malnourished children. Why is it when malnourished children, it turns out around the world, if you just give them food, they don't necessarily get better. It turns out that you need to also think about their microbiome. Why are you doing all this probiotics? Because we have a blunt tool that recognizes that what's in us and on us is very relevant to health. Some of the skin diseases, it turns out, the bacteria on our skin are not so innocent in some of these diseases. And of course, all this is interacting with our immune system. So it really is like turning on the lights for the first time and saying, oh my gosh, there's all these other living things going on, and they're going to be relevant to health and disease. And actually, they may be part of the solution. By the way, they're not just the problem, they're probably part of the solution. And in some cases, therapies are going to be introducing microbes in us and on us to sort of combat and deal with the ones that are misbehaving. And so explosively interesting area, all brought about because of genomic technologies.
So we can all think of ourselves as walking around like Pigpen in Charlie Brown with our little aura. And the thing I take away from this mainly is the incredible hope that this is offering for people in this country and other countries.
And I would really emphasize the international perspective
Yeah,
of this, because
yeah.
so much of what genomics has also done is trying to share data with other countries, embrace collaborations, and also recognize that at the end of the day, if we have these remarkable advances in genomics and only the wealthy countries benefit, only the wealthy people in countries, that will be a mistake. We need to think about how to make sure we don't exacerbate disparities, that we bring these tools and these advances to all people in all countries. And I think that's one of the things I think the community has been very cognizant of, and we continue to sort of really look carefully to what we can do to help that.
So I want to thank you for the talk, and more importantly,